Okay, well, the Secretary of State was talking very much about the present, but he did mention something about the future as well. The part that genomics is going to play in medicine here in this country. And in 2012, you'll remember, the Prime Minister established a rather ambitious vision for genomics in the UK. He committed the NHS to 100,000 sequences of the whole human genome. Now, we've seen an enormous change over time on how much this costs. Remember, it wasn't so long ago that it took 13 years to sequence the first genome, and it cost something like £2 billion to do so. Now, it costs roughly £1,000, and you can do it in a matter of days. So what does this mean? What does this mean to treatment, to your budgets, to your future vision of medical care? Well, we have an excellent person and an excellent panel to discuss this this afternoon. You'll remember Patrick Soon-Shiong from last year. I think Patrick sort of made an entire audience gasp with some of the things that he had to say. And he also tells me that he has two mammoth announcements to make before the NHS Confederation audience here. So those of you who are leaving, yeah, too late now, looking back behind your shoulders. You can come back if you like. Like I said, mammoth announcements. But also, we have with us Sir John Chisholm, Executive Chair of Genomics Confederation England, and Angela Douglas, who is the Programming Director of the North West Coast NHS Genomics Medical Centre. Very excellent people to discuss this. But first of all, please put your hands together for Patrick Soon-Shiong.
Thank you so much, Nita, and what a pleasure to be back, and I'm so glad to be welcomed back. I think today what we're going to do is focus almost as a sequel of what we're going to address here is cancer. And I think we all focus the idea that there's going to be 1 in 2 men and 1 in 3 women in our lifetime with cancer is a frightening thought. Unfortunately, as we understand more about cancer, the complexity of it, the physicians and the oncologists are now faced with what we call a true crisis in terms of not only information overload, but cognitive overload. And we've not won the war on cancer. I think we all understand that it's really been an empirical reactive treatment. We give a drug, select a drug, start again. And when the patient has metastasis, we call this the end, and we end in the patient in hospice. And the reason for that is because, unfortunately, for the last 40 years, we've been under an illusion. We as oncologists and physicians and scientists have been under the illusion of this thing called clonal dominance. We always thought that cancer was a single clone. So therefore, we could nuke it with a chemotherapeutic agent, and that would wipe out most of our normal cells, but hopefully wipe out this clone, and therefore cure cancer. Only in the last few years have we in fact discovered that in fact cancer is a multitude of clones, and in fact it's a multiclonal disease. What do we mean by that? Well, what we mean by that is that there's hundreds and sometimes thousands of clones that have mutated, some asleep, some awake. So trying to hit one out with a single massive chemotherapy or multiple dose of chemotherapy has proven to be futile. So we went to this whole world of targeted therapy, and this is an unfortunate picture of a gentleman with malignant melanoma. And this whole area of targeted therapy became a choice, and the entire pharmaceutical industry is going after targeted therapy. And miracles like this has happened. So here's a patient with a BRAF inhibitor. Very immediately after taking the drug, this is what happened. So if indeed it was a clonal disease, we would have cured cancer. Unfortunately, very soon, this is what happened. So it's taken us 9 years in the industry to develop these drugs and 9 weeks to relapse. So only recently has this new statement come out that we've had this illusion of clonal dominance, and there's an inevitable acquisition of resistance to targeted proteomics. And it's a frightening thought, this new paper that came out, is that resistance is a fait accompli. That is really the time of recurrence is simply the interval required for the subclone to repopulate. I know that's an incredibly sombering thought, and puts us into a position of, are we now lost? Well, we're not, and that's the whole point. It so happens that we have now reached a point of convergence, where I put the slide together for this talk to really try and explain what we've been trying to do for the last decade, or what I call crossing the chasm of scale. We've looked at the population or population health management and tried to bring it to a single patient. And the way we've done that is understood it in terms of tissue. So there's a breast cancer specialist, a lung cancer specialist, and we understood the world of medicine in terms of anatomy and physiology and pathology. In order to handle that in terms of the electronic medical records, we would take these public databases and create electronic medical records around the system, and that's the current state. So that's the current state of our knowledge, and that's our dogma, that we actually have this anatomical method of looking after a patient and following the vital signs. But what's happened now is that we've gone from the tissue to the cell. So there's a whole world of cellular immunotherapy, which you may have heard. And I was just at ASCO, 37,000 oncologists were abuzz about activating T cells. But we've gone beyond the cell, we've gone into the DNA. And Genomics England has taken the very courageous step of saying we need to measure the DNA. But the DNA itself actually encodes for genes which make proteins. So we need to now measure the proteins. But it's not the proteins that we interact with, it's also the peptides. So this area of the anatomy to cellular and molecular biology is now entered. And what's exciting is we've extended all the way from cell to DNA to protein to drug. And that's what I tried to present to you last time, which says we have the capability now to measure what we call this dance of proteins. And our only chance to go from the step of the anatomical current state is to accept this whole world of molecular 21st century precision medicine. So this is what I mean by crossing this chasm of scale. And we need to then therefore take this integrative approach where our current scale of the anatomy and our current EMRs and our current population health and the way we look at population health in our current state has to now in real time for that one single patient be extended to the DNA, to the RNA, to the protein, so that we have what we call this integrative dance of proteins. So this is what needs to be done, this complete analysis in some form of informatics of a single patient, so that we can now move to the 21st century medicine. For the next 15 minutes or so, I'd like to show you how we actually have gone through that. And as you recall, last year I presented this clinical operating system. To take the operating system, it will take electronic medical record merely as an input, but take genomics data, proteomic data, wearable data to the cloud and bring this real-time information to you. And we've now installed this in multiple hospitals in the United States and about to install this in this country. But now let's talk about why does this matter and how do we get there? Well, the whole genome sequencing we're talking about now at Genomics England measures the DNA, and it's 3 billion letters. And these 3 billion letters code for proteins through 20,000 genes called the whole exome. So now that we're going to do this analysis, what's going to happen, we're going to find thousands of mutations. So the question then to you and the doctors are, what is actionable? Well, it turns out that it's not the DNA that's important. It's actually the DNA that talks to the RNA, which talks to the proteins, so that we can get to the protein receptor, which is the target of drugs. So just looking under the DNA is looking under the wrong rock. So I really urge us that it's so critical that you go beyond DNA, and I've just presented that at ASCO. So in order to know which of these 1,000 mutations are actionable, you need to go from DNA to RNA. And if you go from DNA to RNA, we've now established this GPS test, which is a genome proteome seq, a whole genome, whole exome in RNA. in essence, the GPS then creates for us the molecular address of cancer. So we have the molecular address of cancer. We then can bring the Uber of health to this molecular address by targeting this molecular address. So that's the concept, and time doesn't permit me to go through some of these case studies in the last 6 months where we've changed lives of a cervical cancer patient getting a breast cancer drug, of a young patient with a brain tumor we analyzed and found that the patient could be treated with Gleovac, of a female patient who was thought to have pancreatic cancer on a lot of deathbeds. When we analyzed, it found to be ovarian cancer and a complete response with carboplatin. We then found these patients, for example, where we did the protein analysis, and then you see that circle is the element of a patient with a recurrence after chemotherapy of the tumor blocking the esophagus. The patient then found that the patient had a MET protein. We gave a MET inhibitor, and within months, a complete response. So that's very encouraging, except we're facing with this issue of resistance of fait accompli. We hit one clone, there's thousands of other clones. So now I want to introduce you to this whole new concept of what I've called quantum oncotherapeutics. And what I mean by that, I mean you have to play chess with this cancer. You have to recognize its multiple clones. You may have to recognize we can't kill it with a single chemotherapy agent or maximum tolerated dose, and 90% of our treatments today are that. And I struggled explaining this to the community of oncologists, that we need to change the paradigm. Since 2005, 2006, we've started on this program in which we can take low-dose combination therapy and using a nanoparticle of paclitaxel, we now have complete remissions in patients with metastatic pancreatic cancer. And here's another patient with severe pancreatic cancer with completely remissions. So what are we doing here and why? What has happened here is that we've actually enhanced the immune system. And this is a 1908 article by Paul Ehrlich, the Nobel laureate in physiology, that says tumors occur at high frequency in humans, but are kept under control by the human system. It's now a frightening thought, but a real thought, that as we sit here in this room, each of us are creating about 10,000 cancer cells a day in our body. But our own immune system, through what we call innate immunity, a natural killer cell in the body, or a T cell in the body, is actually killing these cancer cells. And the idea is that what's happening in our body is that these cancer cells that are forming have these immune synapses, and the T cells or the effector cells in the body are actually killing them. So I'll show you next year a video produced by the Oxford-Cambridge group of actual T cells, cytotoxic T cells in your body, protecting the body and searching out for cancer cells. And this is an actual T cell, and that blue cell is a cancer cell, having what we call this dance of proteins, and that immune synapse is happening. And these T cells have inside the system these granzymes, these red dots, that are release these enzymes. And through this interaction with this cancer cell, release these, what they call perforin and granzymes, and kill the cancer cell. So this is, in fact, our natural mechanism for killing cancer. So this T cell that I speak about, however, has to learn about this cancer. And this is what's been out there in the industry now, figuring out ways to take the brakes off these T cells, giving them these things called checkpoint inhibitors. And people talk about the concern about the cost. The cost is about $100,000. Now there's the next element of a T cell actually having an antibody attached to it, called a CAR T cell, where an antibody would then target the cancer cell. But that, again, is another huge expense because it's, in essence, a stem cell transplant. On the other hand, in your body and my body, we have this thing called the natural killer cell. And the natural killer cell is sitting out there waiting in its natural state to attack and kill all cancer cells. It's sitting out there waiting to actually release these things called cytokines. And it has in its capacity to release these granzymes. And in fact, it actually has a mechanism to attach itself to antibodies such as Herceptin and kill the tumor cells. So why is this natural killer cell then not active all the time? And the reason it isn't is in order to protect your body from being killed in terms of autoimmunity, it has these inhibitory receptors, which are turned on and then turned on by the cancer cell and put the natural killer cell to sleep. So that is why we have then the proliferation of cancer tissue, is the ability, not only the inability of the cancer cell to die, but also the inability of our own body mechanism to protect ourselves because it activates these inhibitory receptors. And amazingly, since 2005, I came across an amazing killer cell line that has no inhibitory receptors. It, in fact, can be grown in a test tube. It was a cell line from a patient with cancer of natural killer cells and has, in fact, no inhibitory receptors. And the idea was, imagine the day if we could find the molecular address and target these natural killer cells to that molecular address and have these activated natural killer cells kill the tumor cell. So from 2005, this work has been going on, and I'm pleased to announce today that, in fact, this unique natural killer cell, which is a living drug delivered in a blood bag, which could be targeted to a cancer cell and kill the cancer cell, is available, and we're in clinical trials. So we've completed these phase 1 clinical trials. Thank you. We've completed these clinical trials in 40 patients in Toronto, Rush, and we have, these are all early results, 40 patients. This patient now had a complete response with Hodgkin's, non-Hodgkin's, and the patient's now 4 years. And the idea was we could then have what we call this tumor synapse and do this, identify the synapse or the protein by doing the genome proteome sequencing. So the concept then of genome and the proteome and the whole genome analysis is not some hypothetical research project. It's the opportunity to find the molecular address and the thing I call neoepitopes. Because what is exciting we've now discovered is that every cancer for every patient has a unique address, even to the patient's own body. If we can then find that address through the sequencing technique that we've put in place, we can then go search for an antibody and place the antibody onto the natural killer cell. And the next thing I'm about to announce is we now have access to the world's largest library of human antibodies, 10 to the 16, a quadrillion antibody pool that we can go fish and identify the antibody and place it onto the natural killer cell. And we call that a tank, a target-activated natural killer cell. That tank then could be injected into the patient, and it would seek out the tumor, attach to it, and release its granulocytes and kill the tumor. We've shown this in animals, and here's a mouse model with acute myeloid leukemia, the blue, with a tank directed against that cell called a CD33 antibody, and it wipes out these tumors. We've now have an opportunity to take every human being on the planet, identify the synapse, what we call this dance of proteins, through the skill set of whole genome sequencing, whole exome sequencing, and proteomics. And I'm so happy to show you this slide because this is the first demonstration now of a natural killer cell, that yellow, which has been activated with an antibody to target a HER2 positive breast cancer cell. These are normal breast cancer cells. And if it does its job, it would seek out and not touch the normal cells, but go and kill the positive breast cancer cell. And that's what it's doing. It's in fact a serial killer. So you begin to see it has now the capacity to kill these cells. So we're about to then start these phase 2 trials in breast cancer, gastric cancer, lung cancer, in which the ability then to deliver targeted living drugs in the blood bag by taking the activated natural killer cell, identifying the molecular address, creating a bank for that particular patient, growing it up in a blood bag, and it doubles every 48 hours, and placing this then as a transfusion into a patient. We're about to launch these trials in Germany, in the United States, in Spain, and soon in the United Kingdom, so that we can identify patients who could benefit from these based on the sequencing. Which means you need to create what we call a Cancer Knowledge Action Network. And some of you may recall I presented the slide at the last talk in anticipation of the knowledge that each patient generates a terabyte of data. So doing 20,000 patients a day would be 20,000 terabytes a day, which would get you 1,000 terabytes is 1 petabyte. And just to give you some insight, 2 petabytes is the entire National Library of Congress data. 4 petabytes is every spoken word of human, spoken and written word in history. And we already had 4 petabytes of data. So we needed to create this sequencing system that would move data at 8 terabytes a second around the world, which is now in place and in motion. And in fact, moving from the US all the way to the UK. And we have now this Global Action Knowledge Network in place. And I'm about announcing today, I'm pleased to announce with Sir John Chisholm on the stage, that we've been selected as one of the organizations to deliver the bioinformatics report. We've now built a supercomputer system in the United Kingdom that can do the report in seconds. As a consequence of this, the second announcement I'm also making today is that we've just partnered, as of this morning, and this press release has gone out with University Hospital Southampton to create the first genome molecular diagnostic system to bring these clinical trials to this country and to deliver precision medicine for cancer patients using this next generation genome sequencing platform. The idea is for us to actually create a mission control center that will be able to monitor patients and identify not only their molecular address, but take their molecular signature, treat their outcome, monitor their blood signs, the vital signs, and truly have a real mission control and put ourselves, I believe, on the path to the cure. Thank you very much.
to be ambitious enough to take on this degree of linking of data from routine medical care through genomic, and as Patrick has said, it goes beyond genomes. Inevitably, you've got to get the transcriptomes and proteomics in general in the end to get to the proper diagnosis. But it is this digital, data-driven revolution.
But do you have a timeline? I mean, do you have a time scale?
Our project is scaled to 100,000 in till 2017. So that, by the way, is a tremendous ask. If we succeed in doing that, we are well ahead of anyone else.
I mean, this may be a question for either of you, because Patrick, it seems, again, as usual, has set out this sweetie store of things that people will want, desperately want, delicious things in the treatment of cancer. But then it was only a few days ago that a pharmaceutical company, AstraZeneca, was criticising the government, saying, look, there are these excellent treatments for cancer, and they're not paying for them. They are just not paying. Now, how expensive are these sweeties, if you like, and how is NHS England going to pay for them? Maybe this is a question for you.
Okay, so from our perspective, what we do know is that when we treat our patients, even with targeted therapy, we do know that some respond and others don't. So there's an awful lot of money that's wasted on patients who are given drugs and therapies, and even immunotherapy, where they are not going to respond to it. What doing the 100,000 genomes is going to allow us to do is recognize how that, knowing what the actual sequence of a patient's genome is and identifying the actual thing that's gone wrong in the genome, we will then know, are they the sort of patients that are going to respond to this type of treatment, or do we just not give them that treatment at all and give them something else? And by actually precisely targeting the type of treatment that we are going to be giving to our patients, then we'll be able to make the savings from those patients that we are treating that actually will not respond.
So this probably wouldn't fit under a NICE guideline. This is a case-by-case issue. And just on costings, I mean, you are the holder of, I mean, my introduction, it was, forgive me because of time and because the last session went on, but you are the holder of a great number of patents in medical care. You've written more than 100 papers. When it comes to the cost of these treatments, where are we at the moment?
Well, and that's frankly where the next campaign I'm on now, because you're absolutely right. I mean, the checkpoint inhibitors, they estimate $100,000. These cellular therapies, they estimate between $150,000 to $200,000. The chemotherapies themselves, a combination. My vision is to take checkpoint inhibitors, cellular therapy, and chemotherapy, and actually combine them as a bundle in terms of, because that's, by the way, what we need to do, not for cost, but the way you need to attack the cancer cell from multiple fronts. And if you could figure out where pharmaceutical companies not only work together, and they are now what we call basket trials, where they combine their therapies, I think, including generic therapies, 5-FUs, carboplatin, cisplatin, etc., are generic chemotherapeutic drugs. I think you can actually create a cost that is not only sustainable but reasonable.
But when you say reasonable, would it be less than what we're facing with the pharmaceutical costs that already are being resisted by the government right now?
Yeah, and I think part of the issue is exactly as Angela brought up, Herceptin, etc. These are very expensive drugs. And when they're given for patients that actually couldn't benefit from them at all, it's one of these issues. As you know, or may know, that it's the response rate to Herceptin is about 20 to 30%, which means 70% actually do not respond. We've now discovered why. There's a genetic factor called a polymorphism in which the natural killer cells with the other 70% do not bind to this antibody. So if we can find those patients, and then give them the Herceptin, but add a natural killer cell that make it effective, you would reduce the cost, actually.
Okay, so I mean, the cost per unit may not decrease all that much, but you would be giving it out to fewer people because you'd be more targeted.
Right. And I think what Jeremy Hunt just said, you know, the path to better care and lower costs is the same path.
Let us open this up. We've got about 6 minutes for questions. So if you do have, can you paddle people, spread yourself around, and if there's anybody who has a question, please stick your hand very firmly and high in the air. Sir John, just while they're getting to any questions, do you worry that actually, you know, this is such a brave new world? I mean, to me, somebody like me who is a lay person in this field, it is so unbelievably exciting that maybe we are setting expectations that are only going to be disappointed in the fullness of time.
Well, we have to involve people in what we're doing. Be very open about our communication. We are on the edge of science here, but for sure, by the digital revolution, by collecting the data, doing it in a way which we can actually use it, we will progress towards the kinds of understanding which will enable the precision medicine that Patrick has been talking about. Frankly, the way we do medicine at the moment is unbelievably expensive, and that business model cannot work in the long term for everybody. This is the route by which we'll make it an achievable outcome for everybody.
We have a question, number 4, here. Hi.
Thank you very much. Jonathan Inglesfield, GP and Medical Director from Guildford and Waverley. Just thinking through where this takes us in respect of oncology commissioning into the future. It sounds really complex. And I'm kind of thinking that pushes in 2 separate directions. Does it mean that oncology is going to get even more specialized, even more super-centered? Or because there is a kind of science and logic behind it that maybe we haven't had before, does that mean actually you can reduce it down to a smaller, more local level, and actually the clever stuff is done at the sort of, super specialized center?
Thank you. Patrick, can you take that?
I think counterintuitively, it's so complicated that we need to actually create a system from a computer. That's why the title of my talk was Augmented Intelligence, in which really it's beyond the cognitive power of a single human being, whether a specialist or oncologist. The other issue that we need to unlearn is that anatomy doesn't matter any longer, because a breast cancer and a lung cancer and a colon cancer actually may have the same molecular signature. So we need to treat the biology now and not the anatomy, which means that through these decision support trees, which we've now built and deployed across 80% of the oncologists in the United States, could provide not only decision support, but I believe this idea of this mission control center, where you would have a molecular tumor board available remotely in real time, could and should be available, and we're actually creating those kinds of entities in the United States. But because of the internet and the cloud, this is really available on a global basis. So I believe it actually will reduce costs and then make it available all the way down to the general practitioner, and in fact, all the way down to the patient themselves. And I call this patient empowerment as opposed to patient-centered.
One last question. I think number 1, do you have a question? You do. Great.
Thank you very much. It's Sarah Hayes, Director of Public Health of the Health Board in Wales. I'm really interested by your talk. It's opened my mind hugely. And I've got a slightly strange question, which is, would this technique be applicable to treating infections, particularly HIV or TB?
Patrick?
It's a very insightful question. So the natural killer cells is your innate immunity to both viruses and bacteria. So excitingly, that's exactly what these natural killer cells do. So a separate program we have is actually treating Ebola. We've discovered that the way an Ebola actually proliferates is actually kills the natural killer cells. And yet this natural killer cell that doesn't have the appropriate receptor for Ebola can actually fight back. And we've now demonstrated that it can take over some of the cells that are infected by Ebola. Same thing will happen for TB, HIV, influenza. So in fact, it is indeed the innate immunity protective mechanism of one's own self.
Ladies and gentlemen, I can sort of tell when an audience is somewhat stunned into, I can hear your brains whirring from here. We are very lucky that Patrick has spoken to us. He just told me on the way here, he goes, yeah, no, I'm just stopping off, I'm on my way to see Bill Clinton. So he will now go and see Bill Clinton and tell him that he was with all of you. But also let us please thank Sir John Chisholm and Angela Douglas for an absolutely fascinating session this morning. Thank you very much.